So the last speaker is Dr. Charlène Chorao from CSL Behring, and she'll be talking about granted market access to the first gene therapy for hemophilia B. Thank you very much for coming.
So hello everyone, I'm Charlène Chorao. I am a field medical advisor and I work at CSL Behring. I thank you for giving me the opportunity today to tell you, to present you the story of the access of the first gene therapy in hemophilia B, since we recently got an approval for a gene therapy in this disease. So first, I will introduce my company, of course. We are a global biopharmaceutics leader. We develop and delivers innovative and high-quality medicines in rare and serious diseases. And we are present in 4 therapeutic fields, immunology. Okay. I hope this is better. So we are present in 4 therapeutic fields: immunology, hematology, respiratory, and critical care. And we belong to the CSL group, which was founded more than a century ago in Australia. And the CSL group is composed of 3 other companies: CSL Seqirus, dedicated to the vaccines; CSL Plasma, which is one of the greatest plasma collection network in the world; and CSL Vifor, specialized in iron deficiency and nephrology. So in some figures, and the CSL group, this is more than 32,000 of employees in the world, and we have 50 specialties commercialized, distributed in more than 100 countries. So from an R&D side, we have more than 2,000 of employees dedicated to the R&D in the world, and they are spread over 18 centers. You can see the locations of these centers on the map. And you can also see that our company invests heavily in R&D with almost $5 billion, which were invested during the last 5 years. And we have by now 30 specialties under development. Sorry. Here you have the distribution of our 50 commercialized specialties by a therapeutic area. And among them, there is the first gene therapy in hemophilia B. This is in the hematology field. Its name is etranacogene dezaparvovec. So its trade name is Hemgenix, and it is easier to remember, isn't it? So, of course, I will tell you more about Hemgenix later. But before that, I will give you some information concerning hemophilia, its current treatments, and also its unmet needs. So first, what is hemophilia? Hemophilia is a coagulation problem. When you injure yourself and when you have a vascular breach, different physiological processes take place and they allow the bleeding to stop. This is called hemostasis, and hemostasis is composed of 3 stages. The first one, called primary hemostasis, leads to the formation of a platelet plug in the injury site. And this platelet plug is not strong enough to allow the bleeding to stop over time, so it has to be consolidated thanks to fibrin strands. And fibrin is the end product of the coagulation cascade you can see on your right. So this is the second stage of hemostasis, coagulation. And this coagulation cascade, as you can see, this is a complex process. This is different reactions involving different elements, and among the elements there are clotting factors. So when you suffer from coagulation, you suffer from deficiency in a clotting factor. So what I didn't tell you, this is the third stage of coagulation called fibrinolysis. Fibrinolysis, in fact, is a regulation process It limits the extension of the clotting, the platelet plug, and so to prevent thrombus formation, of course. So, as I have already told you, when you suffer from hemophilia, you suffer from a coagulation problem, and you suffer from a deficiency in a clotting factor. There are two types of hemophilia. Hemophilia A, which concerns a deficiency in factor VIII, and hemophilia B, deficiency in factor IX. So when you suffer from a deficiency in factor VIII, this is small, but I have put a cross here. You can produce thrombin, nor fibrin, the end product of the coagulation cascade. And when you suffer from hemophilia B, you have a factor IX deficiency, and you can also produce thrombin, nor fibrin. So when you have a deficiency in factor VIII or factor IX, you can coagulate, and if you can coagulate, you bleed. So a clinical manifestation of hemophilia is the bleeding. And these bleeds can be of traumatic origin, or they can occur spontaneously. That means that you not having been injured before. These bleeds mainly occur in a joint, and they are called hemarthrosis, but they can also occur in muscles or in different organs, like the brain. And in this case, you can guess that this can be more severe and even fatal. So the risk of bleeding is inversely correlated with the level of circulating factor. That means the less clotting factor you have, and the more risk to bleed you have. So here you have an hemarthrosis, which is an effusion of blood in a joint. And this hemarthrosis is often associated with pain, with reduced mobility, and also with swelling, as you can see on the picture here. And a consequence of repeated hemarthrosis are arthropathy, that means destruction, partial or total, of the cartilage in the joint. And in very advanced forms of arthropathy, the patient can suffer from a major disability. So you can see here how severe can be the disease, and you can also see that with this life expectancy curve, which shows you that when the treatments were not optimal at the beginning of the 20th century, the lifespan of patients suffering from hemophilia was only 11 years old. So fortunately, thanks to the current treatments, this lifespan tends towards the normal. Concerning the etiology of the disease, this is a monogenic disease, and it is caused by a mutation in the F8 or the F9 gene. And this mutation can result in a reduction in the levels of the factor VIII or the factor IX, respectively. And both of these genes are positioned on the X chromosome. So hemophilia is an X-linked bleeding disorder. Men are hemophiliacs and women are carriers. And in 30 to 50% of the cases, the mutation occurs spontaneously. So the severity of the disease depends on the reduction of the levels of factor VIII or factor IX, and there are three categories: the severe disease, the moderate disease, and the mild disease. From an epidemiological side, hemophilia is considered as a rare disease since less than 5 people out of 10,000 are concerned. And hemophilia A is really more prevalent than hemophilia B, with almost 80% from hemophiliacs suffering from the form A and 20% from the B form. Just for a little background information, this disease is also called the royal disease, since Queen Victoria of England was a carrier of the disease, and she had 9 children. and some were carriers, some were hemophiliacs. And following the union of her descendants, the disease spread to the other European royal families. So that's why hemophilia is also called royal disease. What are the current treatments? There are two types, two regimens, according to the severity of the disease. So for patients with a severe form, the standard of care is prophylaxis. That means that they receive regular administration of a replacement therapy or other hemostasis products to prevent the bleeding. For patients with a moderate or mild hemophilia, the standard of care is the on-demand treatment. That means that the patient only treats a declared bleeding episode. And on the graph below, these are French data coming from a French registry. They were collected in 2022 for hemophilia B, and you can see that effectively patients with a severe disease are mainly treated with a prophylaxis regimen, whereas moderate or mild patients are treated with the on-demand treatment. Concerning the treatment history of hemophilia, in 1840, the first blood transfusion was used to care a patient, and you can see that more than a century ago, and this is still the same treatment, as well as transfusion of fresh plasma, but these two treatments did not provide enough factor VIII or factor IX to prevent severe internal bleeding. In the 70s, this is the arrival of the first factor concentrates. There are faster treatments, and they offer to the patients the possibility to be treated at home. So this is a good step for the treatment of the disease. In the 90s, as a response to the contaminated blood affair, more secure products are developed, high purity factor concentrates, as well as first recombinant factors. And in 2014, another big step is taken with the commercialization of the first recombinant factors with extended half-life, both in hemophilia A and in hemophilia B. And these factors with an extended half-life offers to the patient the possibility to space the injections. I didn't tell you before, but these patients, when they have to be administered with a standard factor therapy, I mean a factor concentrate or the first recombinant factors with short half-life, this patient has to be administered 2 to 3 times a week, sometimes more, and this is by the intravenous route. So this represents really a big burden, an important burden for the patients. So in 2014, with the commercialization of these factors with an extended half-life, this is really important for the quality of life of patients. In 2018, this is the commercialization of the first non-substitutive therapy. This is only for hemophilia A, and this is with a monoclonal antibody. 4 years later, another big step is taken with the approval of the first 2 gene therapy in hemophilia, one in hemophilia A and another in hemophilia B. This is ours. Sorry for the delay. Here you have the different factor levels profile according to the treatments received by a patient suffering from a severe form. So as I have already told you, when the patient has a severe form, the gold standard is prophylaxis, and the aim of prophylaxis is to maintain the clotting factor true levels above a target of 3 to 5%. In the first case, the patient does not receive any treatment, and as you can see, he has inadequate factor levels all the time, and this patient is not well protected from bleeding, of course. In the second case, on the top, in green, the patient receive a standard factor therapy. This means factor therapy with a short of life, requiring 2 to 3 intravenous administrations a week. And you can see that factor levels spike with every dose and often dip below the target. So what we called the true levels is the level of the clotting factor just before the new infusion of the product. And in the last case, in red, this patient received a long-acting factor therapy. It requires less frequent dosing, as you have guessed, and the clotting factors levels are maintained at or above the target. But this patient still requires regular administration, and you can see that this long-acting factor therapy still results in fluctuating factor levels over time. So you can guess that there are still unmet needs for the treatment of the disease, and since maintaining factor true levels above a target of 3 to 5% requires frequent infusions of the product. So as a consequence, this represents an important burden for the patient, and this may impact his quality of life as well as his adherence to the treatment. So the gene therapy can bring a solution for that, since with a single administration, you have the potential to sustain above target factor levels for months or for years. And gene transfer therapy, as a consequence, was investigated as a novel treatment option a few decades ago. And you have here a timeline for that, with the first clinical trial in hemophilia B published in 1993. This was with a retroviral vector. And 10 years later, another clinical trial in hemophilia B was published using an AAV, and this was by the intramuscular route. The efficacy results were not so good. And since this time, of course, a lot of research works were done. And 20 years later, in 2022, the first gene therapy in hemophilia B is approved, and this is Hemgenix. You have important dates also for hemophilia A on the top of the timeline. So this gene transfer therapy developed the different clinical trials in hemophilia. Uses AAV-based gene transfer therapy. Why an AAV vector? When you are looking for a vector, an ideal vector, you are looking for a vector which is able to deliver the transgene in the appropriate tissues or cells without a substantial toxicity and with a durable effect. So here you have the characteristics of 4 vectors commonly used in gene therapy. The first 2 are non-integrating, the 2 others are integrating. And the AAV is chosen in hemophilia since its gene packaging capacity of 5 kilobases is enough for the F9 gene. For the F8 gene, this is quite different. The researchers had to modify to delete the B domain of the F8 gene. And the AAV is also a good candidate since it presents a low immune response and a long-term expression. So the AAV vector is derived from the wild-type AAV. I suppose that you know everything about the AAV, but just to remind you some characteristics of this virus in a few words. This is a non-pathogenic virus, member of the parvovirus family. Is not able to replicate on its own. And as he has an important prevalence in the environment, a large proportion of the population has been exposed and has developed AAV antibodies, and these AAV antibodies may be sufficient to neutralize a subsequent infection. For example, an administration of gene therapy. So the seroprevalence of these AAV antibodies, in particular the neutralizing antibodies, varies by the geographic locations, by age, and also by the serotype. And here you have an example, a graph, which shows you the difference of the seropositivity between different serotypes. And here you can see that the serotype 5 has the lower neutralizing antibodies, the lower prevalence of these antibodies compared with the others. 13 AAV serotypes have been identified, and they differ from the properties of their capsid. These serotypes present different immunologic properties and also different tropism for tissues, for organs, for cells. So here is etranacogene disaparvovec, Mgenix. We have worked with a serotype 5 AAV. Why the serotype 5? This is because this serotype is particularly effective in targeting liver tissue, and in particular hepatocytes. You have to know that the factor 9 is physiologically produced by hepatocytes. And from AAV neutralizing antibodies, the serotype 5 is also a really good candidate, since he has the lower prevalence, as I have already shown you, and he has a lower titer and also a lower avidity compared with the other serotypes. The transgene we use is the Padua human variant of the gene F9. Why the Padua variant? This is because this is a gain-of-function mutation. This variant leads to the production of a protein really more active, with an activity increase 6 to 8-fold compared with the factor 9 wild type. And the promoter we use is the promoter LP1, which is liver specific, of course. Here you have the clinical development program of Mgenix. And you have to know that before Mgenix, there was a precursor called AMT-060. AMT-060 differs from Mgenix only by one amino acid in the sequence of the transgene. In AMT-060, we have worked with the sequence of the wild type factor 9, and with Mgenix, we have worked with the Padua variant, as I have told you just before. So these two variants differ only by one amino acid, and you have to know that everything else is the same for this gene therapy. That means this is the same capsid and the same regulatory elements which were used. I mean, the promoter is the same, the regulatory sequence is the same, and the terminator is the same. With AMT-060, we have conducted a phase 1-2. This phase 1-2 is a dose ranging study with two cohorts. The cohort 2 is a fourfold higher dose than the cohort 1. And each of these cohorts has included 5 patients. According to the result of this phase 1-2, it has been decided to conduct the further development of the product with the higher dose, I mean those of the cohort 2. So after we developed we conducted a phase 2B, it's still ongoing, as well as the phase 3. In our phase 2B, we have included 3 patients, and in our phase 3, we have included 54 patients. And just to note, during the phase 1-2, patients with pre-existing neutralizing antibodies were not included in the clinical trial, but they were for our phase 2 and our phase 2B and our phase 3. So here is the design of our phase 3 OB. OB, this is for health outcomes with Padua gene, evaluation hemophilia B. This is a phase 3 single dose, multicenter and multinational study, which aims to assess the efficacy and the safety of hemgenix with patients suffering from an hemophilia B, severe or moderately severe form of hemophilia B. And this was only for adults. So before the infusions of hemgenix, these patients had to be on prophylaxis at least for 6 months. This is the lead-in phase. And the primary endpoint of our phase 3 is the annualized bleeding rate, measured from the week 26 to the week 78 after the infusion of the product. And this annualized bleeding rate is compared with the annualized bleeding rate of the patients during the lead-in phase. So you have here the results of this annualized bleeding rate, this primary endpoint. It is achieved with a reduction in the annualized bleeding rate of 64% with hemgenix versus the factor 9 prophylaxis. And not only this primary endpoint, the non-inferiority of this primary endpoint was demonstrated, but also the superiority of hemgenix versus the prophylaxis. And these results were maintained at month 24. Sorry. Concerning the factor 9 activity levels, you can see that after the infusion of hemgenix, the factor 9 levels increase rapidly, and they are in mean at 37% at month 18, and at 37% also at month 24. So these treated patients have now factor 9 levels of almost 37% in mean. We are really far from the 3 to 5% of the target I have told you before for the prophylaxis concerning the true levels. So this is a really good result. And another good result is the sustainability of the activity of factor 9, since at least for these first 24 months, of course, but we have to wait for other data, of course. Concerning the safety profile, no reported treatment-related serious adverse events occurred, and the most frequent treatment-related adverse events were transaminase elevations, and in 9 patients, and all were resolved by corticosteroids treatment. And another important point is that transaminase elevations did not impact the factor 9 expression. Concerning the quality of life, patients experienced a statistically significant improvement of the overall quality of life, and in particular in 4 domains: the feelings, the treatment, the work, school, and the future. So this is counterintuitive, but the lower the score is, and the better the quality of life is. Where do we stand now? We have finished our phase 1/2, and an extension trial is ongoing. We have 3 years of data for our phase 2B, and 2 years of data of our phase 3. 3 articles were published from this clinical trial in really good journals, as you can see, Blood Advances, and the New England Journal of Medicine. And of course, the interim results are always communicated during the congresses. And there is an American Congress at the end of the year, in December. And during this Congress, the results of our phase 3, the 3 years data results, will be presented. So I have told you a part of the scientific story of hemgenix. I am going to tell you quickly a part of this commercial story. So hemgenix was developed by Unicure. Unicure is a company specialized in gene therapy and based in the Netherlands. Unicure owns an innovative AAV-based platform and is able to produce commercial gene therapy thanks to a facility in the US. And according